Hello students, we are today going to talk about another class of vaccines, which is recombinant vector vaccines. And recombinant vector vaccines actually are different from recombinant protein vaccines. If you remember, the recombinant protein vaccine such as our hepatitis B surface antigen vaccine, it consists of a protein only, and that protein has been expressed inside a heterologous or expressing organism like your Speechia or Saccharomyces. That is what actually is a recombinant protein vaccine or a recombinant vaccine, wherein we take out a gene and insert it in a heterologous expression system, and then we produce that protein, and then we inject that protein into the host to generate immunity. However, the concept of recombinant vector vaccine is a little bit different. What exactly is done in a recombinant vector vaccine is that we take an organism which is capable of growing in the host, but is not capable of causing any disease in the host. That means strain, or that strain is some other strain which can grow inside the host without causing any disease. And generally such kind of strains or such kind of organisms are can be bacteria, they can also be viruses. They can grow in the host for a longer duration of time, that is they do a kind of transient growth in the host, and some of the genes inside the genome of these organisms can be replaced. These genes can be replaced with the genes encoding for immunogenic regions or immunogenic proteins of a different organism, of a different pathogen, and then that pathogen's gene is cloned within the host, within the genome of the vector, and then that vector is introduced into the host, so that the vector grows for a longer duration of time inside the host and expresses the gene of interest, and immunity is generated for against those particular genes which are inserted. So such kind of vaccines have gained quite a bit of importance now. There are many vaccines which are under clinical trials for this, but no vaccine is till date available in the market. If you think about the current ongoing COVID-19 pandemic, then in that case we have a vaccine which is being developed by University of Oxford. Now what they are using is a chimpanzee adenovirus vector. That adenovirus vector actually has the capability to grow inside the humans, and now what they have done is they have replaced a part of the genome with a particular gene of new virus or the novel coronavirus, that is the spike glycoprotein that has been inserted into the adenovirus genome, and now that vector is now being is under clinical trials for subsequent use in humans. So in this case we use vectors, we use organisms that have a gene of interest or gene of another pathogen, that could be bacteria or virus, that is cloned into it, and then you can get the response. Another benefit of such vector vaccines is that you are not limited to a one single gene or one single protein. In this case, you can clone a cassette of genes, maybe 1, 2, 3 or 4 genes, as is the carrying capacity of the vector. So you can clone more than one gene inside the vector, and then that can be used to develop a long lasting immunity. The type of vectors that are used in this are both viral and bacterial vectors. Among viral vectors, as I already told you, adenovirus vector is one. Then we have vaccinia virus, that is also one vector that can be used for cloning in the genes from different pathogens, and that can be introduced into the host. Then another kind of vectors which are bacterial vectors, in that it could be, we can use the BCG strain, the BCG vaccine strain. We can remove certain non-essential genes of the BCG, and then we can introduce our own genes of interest. There are some strains of Salmonella typhimurium, the organism that causes typhoid. Those strains have been attenuated, and those attenuated strains of Salmonella typhi can also be used for giving or for providing those kind of genes to the host, so that that actually replicates the host. The vector actually replicates inside the host. The proteins express themselves, inside the host, so that the exposure of the host towards the pathogen, towards the immunogenic proteins, is for a longer duration of time, so that no booster doses will be required, and for a long lasting immunity can be developed because of the long lasting effect, because of the long exposure of the host immune system to these particular pathogenic immunogenic proteins. So another class of vectors which another class of vaccines which is very important is DNA vaccines. People thought, why to use vectors and all those things to deliver actually DNA, why not to directly use DNA. Yes, it has been used at an experimental level, and to some extent there are certain vaccines which are under trials, but no vaccine as such is licensed for use uh, in this involving this technique, or no DNA vaccine is licensed for use. What exactly is done is that we take a plasmid, or a plasmid which is capable of going inside the host, which is capable of replicating inside the humans. That may be a mammalian expression vector, and then you can clone the gene of interest, the immunogenic protein or immunogenic gene inside that vector, and that plasmid is directly injected into the muscle cells of the host or the recipient. Now muscle cells, they take up that particular plasmid, and then they maintain, it has been seen that it can be maintained in an episomal form, that is that extra chromosomal DNA, and it can also be present, sometimes can integrate within the host genome. Now this plasmid, when it gets inside the recipient cells or host cells, it starts expressing the protein. Now this protein actually is an antigenic protein or immunogenic protein. It primes the immune system, both cell mediated and humoral branch, and in this case we can get both TC cell and TH cell activation. If you can see in this diagram, then the plasmid yields the antigenic protein, and then this protein in here it is maintained in as an episomal form. The antigenic peptides can either be used for TH cell or TC cell recognition, and then you have activated B cells and cytokine production, and all the processes, they go on in the same manner, and they can generate immunity which is long lasting. Though they are still under trials, they are not used in any of the vaccines that is available in the market as of today. So if you see, in nutshell, the gene is injected into the muscle cell of the recipient, protein is expressed, which actually induces both cell mediated and humoral response. One benefit is that there is no refrigeration, because DNA can be carried out. DNA doesn't require such kind of refrigeration, and it can be maintained for a long period of time. This is particularly important in third world countries where refrigeration facilities are not that much advanced. So then the limitation is that, of course, it can be used for antigens only, those that are proteins. So that is the limitation of recombinant vector vaccines, DNA vaccines, all of these vaccines. But we cannot say exactly it has a limitation, because most of the antigenic components of a pathogen in most of the cases is a protein, because of its huge variability. So this is in nutshell about vaccines. We discussed in previous videos about, or various previous lectures about the other type of vaccines. So with this we end the topic of vaccines. For more details, you can see the book that I have sent to you. You can see I have already sent you the detailed email, which are the portions that cover this, and that will be all for the vaccines portion. Thank you.